All right, ladies and gentlemen, today we're going to start talking about biotechnology. So, really, how do all of these concepts in biology, from genetics to protein synthesis to stem cells, how are all of these things now, with a basic science understanding, how can we use this to apply that knowledge to improve our lives? That's going to really be the bulk of what we talk about here in this unit. So, first and foremost, let's be honest. Humans have been using their basic understanding of how traits get passed from parent to offspring for generations. We're talking about things like selective breeding, where we have constantly been working to take this big cow and breeding it with this big cow in hopes of continuing to get large cows. Or this plant has this particular leaf shape that we're looking for, so does this one, so let's mate them together and hopefully those offspring will also have that desired trait. That really is this idea of selective breeding. We've been doing it for centuries. Farmers, ever since really the dawn of agriculture, have been taking plants, crops, animals, livestock, breeding them together in hopes of getting traits continuously in the offspring that they want. Now, that being said, go back and remember your genetics. If that trait is recessive, then their likelihood of getting that trait continuously in the offspring is probably pretty decent. But if it's a dominant trait, you're going to constantly end up with those situations where you get those recessive characteristics to sort of pop up every now and again. And then let's also keep in mind that not every characteristic in every organism is inherited the way Mendel saw with his pea plants. Not everything is dominant or recessive. There's a lot of gray area in the way that traits get passed on. So as a result, the selective breeding, while we've gotten pretty good at it, is definitely not a guarantee. They're not always successful, and even if we are, it's going to take time to build up those purebred lines when we're just pulling these organisms out of nature. So that's where biotechnology really has come in as we've continued to strengthen our understanding of molecular biology. So, when we talk about biotechnology, just by a definition standpoint, it's really the use of organisms to perform some sort of task that we as humans have decided is worthy of our time and endeavors. Now, a lot of that biotechnology has really come from our understanding of genetics and from DNA replication, protein synthesis. So, we're really going to kind of start off today talking about biotechnology in that sense, using the genetics and the DNA as a way to get organisms to do what we want them to. That being said, that term biotechnology really has a much broader context if you want it to. We're talking about biotechnology, and you can throw selective breeding in there. We can also add things like our understanding of cells and talk about stem cells and gene therapy and all of that kind of stuff, and that will continue to come up as we work through this unit. For today, though, let's focus on the sort of DNA side of things. So, when we look at biotechnology from that side of the coin, which is pretty common at this point, we're talking about things like genetic engineering, which really is a technology within biotech in which we take the genome, and remember, genomes are the collective traits, the collective DNA as a whole in a living cell, and we modify it. And we're modifying it either for usually medical or some sort of industrial use. The way that that works is what we're going to dig into today. And genetic engineering overwhelmingly depends on recombinant DNA. So, what does that mean? Well, it's recombining DNA. We're taking DNA from two or more organisms and splicing them together, putting them together, and creating these genetically modified organisms, or you may also come across the term transgenic organism. So, why do we bother doing this? Well, genetic engineering as a whole allows us to take huge advantage of the fact that DNA to RNA to protein, that sort of process of transcription and translation, the way that your cells express their genetic information to make some sort of protein product, is across the board the same. It works the same way whether we're talking about an E. coli bacteria, whether we're talking about you as a human, whether we're talking about corn plants in the field or the pig in the barn. Every organism on Earth uses the same basic principles of taking that DNA, creating messenger RNA from it, and then using the ribosomes and that messenger RNA code to then produce proteins. So it's really easy to now, as we're starting to understand that process and how your cells use that information, taking a gene and moving it into another organism, and that new organism will still be able to use that information just as its original host did. So we use genetic engineering to make things like insulin. Okay, insulin is a protein in your body that gets produced as a result of helping to keep your blood sugar in check. If you have diabetes, however, your body isn't naturally good at producing that insulin. So historically, where did diabetics get their insulin? Well, we got it out of pigs. So those pigs, now we required space and food and time to raise those pigs, to then harvest that insulin, to isolate it, to give it to the patient. And because it's not human insulin, your body has the potential to recognize it as not human, and as a result, destroy it, reject it. Your immune system recognizes that it's not human and gets rid of it. So there's that aspect. There is also a lot of ethical issues. There are financial issues with using that insulin from pigs, religious issues. If you are a very strict, kosher, Jewish practicing person, pigs aren't kosher, and so you weren't necessarily allowed, based on your religious beliefs, to go ahead and use that insulin. So, nice thing is that genetic engineering now has made all of those ethical issues, those religious issues, even a lot of the financial issues to kind of disappear, because now we can genetically engineer bacteria to do the production of that insulin. And we're going to use that insulin example as we work through this today. It's cheaper, it's faster, and it's human, which is awesome. We can also use genetic engineering to make any other organism with a specific trait that we're looking for. So, talking about crops, we're looking at corn resistant drought resistant corn, or corn that is what we call Roundup ready. And Roundup is a pesticide, so we can spray that on our crops and not have to worry about the pests that would normally ingest the corn, because now they're going to ingest that gene and I can use the pesticide and I don't have this issue. So there's a whole host of awesome examples out there as to the way that genetic engineering has been incredibly helpful. So, although we're going to focus mostly today on the recombinant DNA piece and just how this is all possible, genetic engineering then also can layer on top of things like DNA fingerprinting, the GMOs, specifically the genetic engineering, the cloning, the gene therapy. And so these are some other topics that we're going to hit as we continue to work through this unit. So first and foremost, let's really talk about just how the process of genetic engineering works. And the first thing that we need to understand is that in order for genetic engineering to be able to do its job, I have to be able to get that gene of interest. So let's just say we're using that human insulin gene as our example for this. I need to be able to get that human insulin gene out of a human and into some other cell. In this case, we're usually looking at bacteria. So what we're talking about are these vectors. It's some sort of agent that's going to carry the gene that I want into another cell that isn't where it originally came from. So those vectors can be things like viruses. We're going to bring that back up when we talk about gene therapy. It can be yeast. We're going to focus today on how we use bacterial plasmids as vectors. So let's talk about what a plasmid is. So if I take a look down here, here's my bacterial cell. And if I look, remember these guys are prokaryotes, so no nucleus, but they do have a single circular chromosome in the center of their cell, but they also have these extra little rings of DNA. And those plasmids are kind of interesting. I like to think of them as a way to give bacteria sort of like superpowers. So remember, these guys are prokaryotes, so there's really no way for them to be able to get variation into their population because they're asexually reproducing. Although here's the caveat. The reason bacteria have managed to exist on this planet for pretty much the entirety of life is because they found ways around that lack of genetic variation as a result of their asexual reproduction. How did they do it? Well, most of it's coming from plasmids. And so these plasmids are separate rings that are not part of their main chromosome, but they're still DNA. So those DNA plasmids can still produce information for this bacterial cell to produce new proteins. So it can still go through transcription and translation. They can also self-replicate, so I don't necessarily need the bacterial cell to go ahead and go through that replication, that binary fission process. I can just make new copies of these plasmids. Bacterial cells are really good at sharing those plasmids with each other through a process like conjugation that we talked about. They're also really good at transforming themselves. So we talked a little bit about transformation back in the beginning part of the DNA unit. And so they can pick these plasmids just straight out of their environment, which is really what is going to allow this genetic engineering process to work. So how do we use these plasmids? Well, we've got to do sort of these basic steps. One, I've got to remove that plasmid from a bacterial cell to begin with. And even that step has gotten sort of circumvented because we can actually engineer these plasmids now in a lab as opposed to having to isolate them initially, which is pretty cool. Once we get that plasmid out of the bacterial cell, I also have to extract my desired gene from wherever it's coming from. So if we stick to that insulin gene, that means I need to be able to know where it exists in a human cell's chromosomes so that I can go in, I can cut it out, and now I have that human insulin gene, that set of A's, T's, C's, and G's that I need. So I've removed the bacterial plasmid, I've removed that insulin gene from a human cell, and now I'm going to put them together. All right, and we'll talk a little bit about how that works, but I'm going to create that recombinant DNA. So I'm going to take bacterial plasmid DNA plus human insulin DNA, put it together, and create that recombinant DNA. That recombinant DNA, or in this case, that recombinant plasmid, now carrying both bacterial DNA and that human insulin gene, get put back into your bacterial cell. We'll use that transformation process where we'll encourage them to take it up out of their environment. And then once that recombinant DNA is in fact in that bacterial cell knows what to do with it. Its DNA is DNA is DNA. It doesn't matter where it comes from. So they'll be able to run through transcription and translation to now express that additional human insulin gene, and off they go starting to make human insulin. The other cool thing is because these bacteria take up this gene, as soon as they go through their binary fission process, so long as we keep them happy and they want to hang on to that plasmid, they'll go ahead and copy that plasmid right along with the rest of their DNA. So now I may have transformed one bacterial cell with that recombinant DNA that had the human insulin gene, but so long as I keep them in the proper conditions to want to replicate, I'll now have two cells that have that human insulin gene. Allow them to do it again, and now I've got 4 cells. So you quickly, within 20 minutes, double your generation if we're talking about something like E. coli. So after a few hours, you've got hundreds of cells that are now hanging on and expressing that human insulin gene. That's this idea of gene cloning. And we use it not only for things like insulin, which is where that synthetic hormone idea is coming from, but we can use this to make antibiotics, we can use it to make components to vaccines. Really, the ability for this process to work is virtually endless, so long as we know what the code is in the DNA to make the product that we're after. So one of the workhorses of genetic engineering is this thing called a restriction enzyme. So restriction enzymes are really cool. So if you remember, I think I mentioned this back when we talked about DNA, restriction enzymes are actually naturally produced by bacterial cells as a way to be sort of their immune system. The restriction enzymes are enzymes, so proteins, that specifically cut between the sugar and phosphate bonds in your DNA. So remember, DNA, you've got your double helix, you've got your sugars and phosphates in the backbone, and then your nitrogenous bases, which are your A's, T's, C's, and G's down the middle. So restriction enzymes will come in and it will recognize a certain code. In this case, if I look at this one, the recognition site is always between a G that has two A's, a T, double T, and a C after it. So it looks for that very specific sequence and it cuts the sugar phosphate bonds here. And then on the reverse sequence, it's what we call a palindrome, so it's opposite. And so it'll cut here. So it'll splice this guy open and create what we look at here. So those restriction enzymes, we're using them as a molecular set of scissors for all intents and purposes, but they're naturally occurring in bacterial cells as a way to chop up viral DNA so that they don't get infected with a virus, which is pretty cool. So we've taken their own natural defense system and now used it for our own purposes. So we're using them as a way to create what we call sticky ends in your DNA. So these sticky ends are these single-stranded DNA strands, real short little strands here. And remember that between your nitrogenous bases, they want a hydrogen bond, so they don't like kind of just hanging out here all by themselves. It's not very stable. So if I use the correct restriction enzyme on my plasmid ring, it's literally taking that circular DNA and opening it up. I'm flattening it into a straight line. But now I've got sticky pieces on either end. So it's like taking a taped ring together, taking it apart, but allowing the tape on either side so it's still sticky. I'm going to do the same thing to that DNA fragment that I wanted. So if this is my insulin gene, all right, I'm going to take the same thing. I'm going to use another restriction enzyme that gives me complementary sticky ends so that they base pair together so that when I stick my insulin gene in the test tube with my restricted cut plasmids, they want to stick together. They want to base pair together. And so we'll allow this process to happen, which reforms the plasmid. I'm putting the plasmid back together, but now I have this extra piece in the middle. I have that insulin gene within it. Now, there's still breaks here in these backbones, so you've got that DNA ligase to come in just like we looked at in DNA replication. Remember that DNA ligase comes in to seal up the backbones in your primers once they get removed, as well as within the backbones of all of those Okazaki fragments. So we'll seal everything up, and now I've got a complete ring of DNA that's able to go back into a bacterial cell, capable of going through transcription and translation to make that protein product. And in our example right now, we're talking about that insulin. So essentially at this point, I've created recombinant DNA. So here's another way to look at what we just talked about. Here's that bacterial plasmid. Here's my human chromosome and my insulin gene. So I'm going to use that restriction enzyme to cut open that ring. So I've created those sticky ends here. And I'm going to use the same restriction enzyme in my human insulin gene to create sticky ends on that gene as well. Now you've got to pick the right restriction enzyme to be able to create sticky ends here and here so that they'll ultimately seal back together. This is this idea of gene splicing. Splice means to cut. So I'm cutting them open with that DNA. Once I do that, that human insulin gene can then be inserted into a test tube with a bunch of cut bacterial plasmids, and then we'll use that DNA ligase, we'll seal everything back up, and now I've got that recombinant DNA, or that rRNA, if you ever see it shortened like that, or rDNA, excuse me. rDNA is recombinant DNA. So I've now created that. Once I get that into a bacterial cell, or if I'm doing this with some other plant, that's another common way for this to work, or even a virus at that point, I've now created some sort of transgenic organism. I'm taking genetic information and I'm transferring it. That's where that term kind of comes from. I'm transferring it into another organism that doesn't naturally have that gene. So that recombinant DNA then, here's where I create that transgenic organism. I'm allowing it to insert into a bacterial cell in this case, and then I let that guy go ahead and reproduce. So I've got one bacterial cell that may have taken up that recombinant DNA, and now I let it go through binary fission, make copies of itself, and it's going to make copies of that recombinant plasmid right along with it, which is great because now I allow that bacterial cell to do most of the work for me. I don't have to make 10,000 recombinant E. coli in order to get enough insulin production to be able to sustain and utilize for an individual who has diabetes. Instead, I can transform, say, maybe 10 bacterial cells, allow them to go through cell division, reproduce that recombinant plasmid right along with themselves, and they do most of the work for us.
Recombinant DNA technology is the process by which scientists can modify the genome of an organism. This process can be used to insert desirable genes or remove undesirable genes in order to improve the organism. To be able to insert a new gene into an existing chromosome or plasmid, the gene must first be isolated from its source organism's DNA. Source DNA is enzymatically broken into small pieces. A restriction enzyme that's able to recognize a particular nucleotide sequence near the gene of interest is added to the mixture of DNA pieces. The restriction enzyme cuts the DNA at the specific recognition sites near each end of the gene of interest. Note that the fragment that results has single strands at each end. These are termed sticky ends and will be the place where this fragment attaches to the recipient cell's DNA. The recipient DNA, often a plasmid, must also be cut to allow a place to insert the desired gene. The same restriction enzyme cuts this DNA in the same manner, opening up a place for the sticky ends of the donor DNA to attach. DNA ligase attaches the complementary sticky ends to one another, producing a recombinant DNA molecule. The recombinant DNA molecule is inserted into a host cell, where the gene is copied many times, or it's transcribed and translated, producing many copies of the desired protein molecule.
So, why is it really important that we are able to do this at this point? Well, so we've got the insulin there, right? So I can make recombinant pharmaceuticals, which is a great example. We can also use it to make vaccines. We can use it to get all sorts of traits into crops to make them better, drought resistant, heat resistant, bigger fruits, pretty much you name it, we're able to do it. Plants that are able to produce either their own insecticides or pesticides, or that pesticide or insecticide doesn't destroy them, but it only works on the pests themselves. We can do it in order to do gene therapy. So being able to give somebody the correct functional healthy copy of a gene as opposed to the mutated disease causing one that they were born with. We can do vaccines. Really, this is just a really small list of what recombinant DNA allows us to do, but it really is ultimately the workhorse to most of the biotechnology that we see out there today. So if you take a look, this is just a great sort of real quick overview of how this works, right? So here's my bacterial cell, pull that plasmid out, my donor cell with my gene, pull that out. I've got to use those restriction enzymes to take that gene out of that donor cell's DNA, use the same restriction enzyme to cut the plasmid open, allow those sticky ends to now stick together to create that recombinant plasmid, use the DNA ligase to seal it back up, and then allow that recombinant DNA to get back into that bacterial cell so that this guy can then go ahead and reproduce, create more copies of that gene while also producing the protein that is on that plasmid as well.